Hello, everyone. My name is Hiro Patel, and welcome to my Tech Talk. So, today we will be diving into the world of microbiology and specifically types of mediums. So, let's get right into it. An overview of what we will be discovering today. So, first, we'll discover what exactly a culture medium is. Then, we'll go on to discuss the difference between an enriched medium versus a selective medium. Later, we'll see what they're each used for, what they're each made of, and what makes a good medium. So, what is a culture medium? Well, a culture medium is used in a lab setting. It is a liquid or gel designed to support the growth of microorganisms and allow them to proliferate. There are different types of media suitable for growing different types of cells, depending on one's need. As you can see, there is an image of blood agar selective medium preparation, and this is one of the mediums that we will dive into later on in the presentation. So now we will be discussing the difference between an enriched and selective medium. While both of them are used to grow colonies, there is a very distinct difference between the two. An enriched medium is usually solid. It contains nutrients required to support the growth of a wide variety of bacteria, even those that are normally hard to grow. On the other hand, a selective medium contains the nutrients to allow for specifically selected bacteria to grow. So what are these mediums each used for? The enriched medium can grow a variety of organisms, including fastidious ones. A fastidious organism has a very complicated nutritional requirement. The bacteria cannot make certain nutrients, so they require them to be added to the enriched medium to allow for their growth. In a selective medium, if a sample contains a mixture of various bacteria species, this method will help isolate a specific type on an agar plate. What are these two mediums made of? Generally, a good medium intended for cell growth would consist of the basic elements that a cell would need to proliferate. A general purpose media consists of peptone water, nutrient broth, and nutrient agar. For enriched medium, in addition to the basal medium, lots of extra nutrients, growth factors, and vitamins can be added in the form of blood, serums, even egg yolk. For a selective medium, it really depends on what type of bacteria you are selecting for. For example, for McConkey's selective bacterial growth medium, it'll only allow the growth of gram-negative bacteria. Additionally, selective media such as TCBS agar, which stands for thiosulfate citrate bile salt sucrase agar, is used for isolating V. cholerae from fecal specimen because they have an elevated pH of about 8.5, which inhibits the growth of most other bacteria. Additionally, if you know that a microorganism is resistant to a certain antibiotic, this can be used to their advantage. For example, if the organism is resistant to ampicillin or tetracycline, then that antibiotic can be added to the medium to prevent other cells from growing that do not possess this resistance. Now that we've learned a little bit more about each of these mediums, what makes a good medium? Well, in general, it depends on what your purpose is for the experiment you are trying to conduct. An enriched medium would be a good one if it fulfills its function of allowing for a wide variety of organisms to grow. This means that it should include all the nutritious contents needed, even for fastidious bacteria. For example, for the blood agar medium discussed earlier, it consists of the basic elements like a little bit of peptone, which is a protein digest, yeast extract, agar, some sodium chloride, and of course the main ingredient being 5 to 10% of sheep blood. And here you can see an image of some colonies grown on blood agar. A selective medium, a good one, fulfills its function of ensuring that the selective bacteria is the only bacteria that is able to grow. So it should be very specific and not broad spectrum. For example, mannitol salts agar medium has a high concentration of sodium chloride, up to 7.5%. So it will only allow the growth of halophilic, salt-loving bacteria, such as streptococci. Other types of bacteria would not be able to survive in these conditions. And that concludes our presentation for today. I hope you learned a little bit more about the world of microbiology, enriched mediums, and selective mediums. I hope you have a wonderful day. Thank you for listening.